Welcome back.
Imagine, imagine for a second that you are just standing in your bathroom.
Okay.
You gargle some salt water, you spit it into a cup, and then you mix it with just a little bit of soap and some rubbing alcohol.
Right. Classic high school biology experiment.
Exactly. And if you do this, you are going to get this sort of gooey, clumped mass floating right at the top of the cup. And that right there is human DNA.
Yeah. Mostly yours. Maybe a little bit belongs to the bacteria living in your mouth.
Yeah, gross but true. But here's a really crazy part about that. Even if you took that gooey mass and you, I don't know, put it under $1 million electron microscope, looking at it is completely useless.
Totally useless.
You cannot actually read the genetic code that makes you you.
And that really is the ultimate paradox of human biology. For the vast majority of our history, our own genetic instruction manual has been entirely illegible to us.
We knew it was there.
We knew it was there, but yeah, but it was just this lockbox. Think about trying to find a single specific mutation, like the one tiny genetic typo that causes sickle cell anemia.
Right.
You're looking for a single letter change among 6 billion letters in the human genome. Finding that needle in a haystack was, at the time, basically a statistical impossibility.
Trying to find a specific DNA sequence is like reading a license plate on Interstate 5 in the dark from the moon.
It's a great visual. It's just impossible.
So welcome to today's deep
Modern
dive. Our mission
historians-
today is to explore a fascinating video transcript about the invention of the DNA photocopier.
Yes.
It is the wild, entirely unexpected story of a technology that literally changed the modern world and how humanity finally learned to read its own genetic code.
Okay, let's unpack this. Because the guy who figured this out was not your typical polished lab coat wearing scientist. Far from it. I mean, the central figure in this story is a man named Kary Mullis. And to understand the breakthrough, you really have to understand the mind that produced it.
And Mullis is just a bizarre character to find at the center of one of the 20th century's greatest scientific breakthroughs. We were talking about the 1960s at UC Berkeley.
Right.
He is a biochemistry student, but he is barely going to class because he is, he's too busy experimenting heavily with LSD.
Yeah, he was very much a product of that time and place.
Definitely. And he fills his PhD dissertation with so many jokes that the committee actually refuses to approve it until he cuts out all the wacko stuff.
I made him edit it down.
Right. And yet his brain operates on this completely different wavelength because at age 22, he writes a paper chronicling the entire universe from beginning to end, sends it to the prestigious scientific journal Nature.
And they actually publish it,
Yeah.
which is getting published in Nature at 22 is incredibly rare.
It's unheard of.
But after graduating, his career doesn't exactly skyrocket into elite academic circles. It kind of meanders.
Yeah, he bums around for a while.
He does.
Yeah.
He gets a job in a cardiology lab, but he quits because he is too horrified by the number of rats being killed for the experiments.
Which is fair.
Yeah. Then he tries writing fiction. He even ends up working in a bakery for a time.
And the bakery is actually where destiny sort of knocks on the door. A chance encounter with an old acquaintance completely shifts the trajectory of his life.
Right. A scientist named Tom White walks into that bakery.
Ah.
And White knew Mullis from Berkeley and knew he was actually an excellent synthetic chemist.
Partly because Mullis had been synthesizing hallucinogenic drugs back in college.
Exactly.
Mhm.
So White recommends Mullis for a job at a company called Cetus. The year's 1971, and Cetus is one of the very first biotech startups in the world.
Okay.
This is an era when scientists are just starting to figure out how to physically manipulate DNA.
And to understand what Cetus was trying to do, we have to talk about nanoscopic scissors.
Yes. So that same year, researchers at Johns Hopkins were studying bacteriophages. Those are viruses that attack bacteria. And they noticed that some bacteria had evolved these defensive molecules that act like microscopic scissors. They are known as restriction enzymes.
And what do?
They basically scan the virus's DNA, look for a specific sequence, and just snip it into useless pieces. Scientists realized they could extract these enzymes and use them to cut any DNA, including human DNA, wherever they wanted.
Okay, so Cetus wants to use this new scissor tech to create commercial DNA tests for hospitals.
Right.
The goal is to quickly diagnose something like sickle cell anemia. But the existing method to do this was just agonizingly slow and, frankly, really dangerous.
It was a process called the Southern blot. Let's break down how highly inefficient this was without getting bogged down in the textbook jargon.
Please.
First, you use those restriction enzymes to chop the DNA sample into fragments. Then, you spread those fragments out by size using an electrical charge.
Like pushing them through a gel.
Exactly, gel electrophoresis. But you still can't see the specific mutation. You just have invisible DNA sorted by length.
So you have to find a way to make the specific mutation light up.
Which meant heating the DNA to over 90 degrees Celsius to break the bonds and unzip the double helix.
Okay.
Then, scientists would build custom short pieces of synthetic DNA called probes. These probes were designed to be a perfect complementary match to the mutation they were looking for.
And how did they see them?
Well, to make them visible, they made these synthetic probes radioactive.
Oh, wow.
Yeah, you watch the unzipped sample with the probes. If the mutation is there, the radioactive probe sticks to it. Finally, you check the sample for radioactivity to get your result.
And this took days, right? Sometimes weeks.
Weeks, easily.
You have lab technicians handling highly radioactive material, and the signal you got at the end was often incredibly faint. It was completely unviable as a fast commercial hospital test.
Not scalable at all.
Not at all. And here's where Mullis comes back into the picture. Because he wasn't the brilliant mind designing these cutting-edge diagnostic tests. His job was the grunt work.
Right, he was the guy synthesizing those tiny radioactive probes day in and day out.
It was slow, repetitive, and extremely boring manual labor. Think about how much of his cognitive bandwidth was being wasted just mixing chemicals.
And because his brilliant, chaotic mind was so chronically understimulated, his personality really started to grate on his coworkers.
That's putting it mildly. The guy who was frankly a nightmare to work with.
Yeah, he was tough.
He picked fights with receptionists and security guards. He was a notorious womanizer who constantly hit on people at the office. At one point, he even threatened to bring a gun to work.
It was a toxic situation.
He's completely alienated from his colleagues. Nobody wanted to be in the same room as him.
If we connect this to the bigger picture, something crucial happens next. A turning point that is incredibly relevant to you, the listener, living in today's highly automated world. CETUS brings a new machine into the lab.
Right.
This early piece of automation could do a month of Mullis's tedious synthesis work in a single day.
It automated his job.
Exactly.
But instead of being laid off, he suddenly has all this free time and a surplus of these little DNA snippets to just play around with. His mind is finally freed from the busy work.
Yes.
And since nobody at the office actually wanted to be around him, he started spending his weekends driving out to his cabin in Mendocino County.
It was on one of those Friday night drives in the spring of 1983 that everything changed.
He is driving, and he just lets his mind wander. Because of his past experiences with LSD, his brain was uniquely accustomed to visualizing incredibly complex, abstract things.
He isn't high on this drive, but he later noted his mind had learned how to get to that state naturally.
Yeah, he could literally visualize DNA molecules unwinding in the air around him. He saw pink and blue electric molecules hovering over the mountains.
He was thinking about the fundamental problem with the southern blot, that faint, nearly unreadable radioactive signal, the whole issue of trying to read that license plate for the moon.
And his epiphany was so beautifully simple. He realized instead of trying to invent a more powerful telescope to read that single license plate,
What if you just copied the license plate?
exactly. What if you made 1 billion copies of it and covered the Earth with them? Then you wouldn't need a telescope at all.
This is the birth of the DNA photocopier.
Right.
To understand the genius of what Mullis visualized on that drive, we have to look at the 3-step mechanism he conceptualized.
Let's do it.
Step 1 is unzipping. You heat the DNA sample to over 90 degrees Celsius to break those bonds, splitting the double helix into 2 separate strands.
Step 2 is targeting. Remember those little snippets of DNA he was making at his boring job?
The probes.
Right. He realized he could use them as primers. If you know the sequence you are looking for, you create primers that pair exactly to the start of that specific sequence on the unzipped strands.
And then step 3 is the extension. You introduce DNA polymerase. This is a naturally occurring enzyme responsible for replicating DNA in every living cell.
Okay.
The polymerase finds those primers and starts grabbing loose nucleotides floating in the mix, attaching them to build a perfect complementary strand.
But wait, if the enzyme just starts building, wouldn't it just keep copying the rest of the DNA strand forever? How did he isolate just the tiny piece he wanted to copy?
That is where his understanding of molecular geometry comes in. DNA strands have a strict chemical direction, like a one-way street.
Right, the 3' and 5' ends.
Exactly. The copying enzyme physically requires a specific chemical hook, an OH group on the 3' end to attach the next piece. It avoids the phosphate on the 5' end. So it can only ever build forward, never backward.
It's a one-way street.
Yes, most people might see that as a limitation. Mullis saw it as the key to boxing and the target.
So he points two primers right at each other on the opposite unzipped strands.
He did. The polymerase extends both primers toward each other, isolating and copying only that specific segment in between.
That is brilliant.
You do those 3 steps, unzip, target, extend, and you turn one copy of the DNA segment into 2. Then you do it again and you have 4, then 8, 16, 32.
It's an exponential explosion. If you repeat that cycle just 30 times, you go from one single invisible strand of target DNA to over 1 billion identical copies. You've amplified the signal so much that it is impossible to miss. Mullis had just invented PCR, polymerase chain reaction.
And he is so stunned by the math that he pulled his car over and scribbled the entire concept down on the back of a gas receipt.
I love that detail, but having a brilliant idea on a gas receipt is very different from proving it in a laboratory.
No, reality hits hard on Monday morning.
It really does. He bursts into CETUS, pitches his DNA Xerox machine, and people literally walk out of the seminar.
They think it's way too simple.
Right. They figure if it works, someone would have tried it already. And when Mullis actually tries to run the experiment on human DNA, it completely fails.
Human DNA was just too large and complex for his initial clumsy attempts. And Mullis wasn't helping his own case.
No, his lab work was incredibly sloppy. He wasn't running control groups. He wasn't doing repetitions. He was rapidly losing all credibility to the point where he got into a literal fist fight with a colleague over his data.
Everyone wanted him fired.
But a few key people at CETUS, Tom White, Norman Arnheim, and Henry Ehrlich, they realized the concept itself was sound.
They put Mullis on a 1-year probation and brought in a team of disciplined technicians to actually make it work.
Which was exactly what was needed.
Right. And by the spring of 1985, by scaling down to a tiny 25 base pair fragment of bacterial DNA, the team finally proved PCR was real.
But there was still a massive, seemingly fatal flaw in the process. We have to go back to the unzipping problem from step 1, heating the DNA to over 90 degrees Celsius.
This is a huge roadblock.
Yeah.
Yep.
The DNA polymerase they were using to copy the strands was extracted from E. coli bacteria. E. coli dies at around 50 degrees Celsius. So every single time they heated the test tube to 95 degrees to unzip the DNA, they completely destroyed the polymerase enzyme.
Which meant that for every single one of those 30 cycles, a technician had to stand there, wait for the tube to cool down, manually open it, and drop in fresh polymerase.
It was tedious, expensive, and completely ruined the dream of a fully automated process. So how do you get around that? You can't just change the laws of thermodynamics.
You don't. You look for something in nature that already beats the heat. And to find this incredible detour back to 1964.
Okay, set the scene.
A microbiologist named Tom Brock and his undergrad student Hudson Freeze take a trip to Yellowstone National Park.
Yellowstone.
Yeah. They are looking at these boiling hot springs, which are full of these vivid yellow and orange colors. At the time, the scientific consensus was absolute. Nothing can live above 60 degrees Celsius. The water is literally boiling.
Right.
But Brock looks at the colors and thinks it might be biological.
So they take samples. For days, the undergrad sees nothing. But on the 4th day, he looks under the microscope and sees worms and bacteria thriving in boiling water.
They had discovered a new organism, which they named Thermus aquaticus, or TAC for short.
TAC.
This raises an important question. How does a bacteria survive boiling water?
Right.
The answer is that its internal enzymes had evolved to function perfectly at extreme heat. For 2 decades, this was just a neat bit of trivia stored in a microbial database. Nobody thought it had any practical application.
Until 1985, when Kary Mullis is hunting for a heat-resistant polymerase, he stumbles on this 20-year-old discovery. The team isolates the polymerase from the TAC bacteria and drops it into the PCR machine.
And the results weren't just good, they were perfect. The CETA scientists who purified it called it the Holy Grail. TAC polymerase easily survived the 95-degree unzipping phase, but it did something else entirely unexpected.
Because it loved the heat, they never had to lower the temperature of the reaction below 50 degrees.
Exactly. With the old E. coli method, they had to drop the temperature down to 30 degrees. At that low temperature, the primers would sometimes accidentally stick to the wrong parts of the DNA, creating messy background noise.
But with TAC, the temperature stayed high enough that the primers only had the kinetic energy to bond to a 100% perfect match.
It completely erased the background noise. You could literally set the machine, walk away, and come back 2 hours later to 1 billion perfectly clean copies of whatever DNA you wanted. They had done it.
Which is just incredible. And this is where the corporate drama really kicks into high gear. CETUS knew they had world-changing technology on their hands, and they needed to patent it immediately.
Other companies were sniffing around. If anyone else published the concept of PCR first, CETUS would lose everything.
So the team begged Mullis to write the paper since the core idea was his. But he procrastinated.
As he tended to do.
Yeah. With time running out, the rest of the team was forced to write it themselves. The very first paper on PCR was published in Science magazine in December of 1985, and Kary Mullis was listed as the fourth author.
He was absolutely furious. He felt his solo genius was being stolen. By the time he finally wrote his own paper, top journals like Science and Nature actually rejected it.
Wow.
The relationship was completely poisoned, and shortly after, Mullis quit CETUS.
But PCR went on to conquer the globe. It became the literal DNA photocopier in every biology lab on Earth. The impact is staggering. Think about forensics.
Oh, absolutely.
Imagine you are in prison for 20 years for a crime you didn't commit, and a single hair follicle left at the scene, multiplied 1 billion times by PCR, definitively proves your innocence. It has freed hundreds of wrongfully convicted people.
It also allowed doctors to detect cancers at incredibly early stages. It allowed scientists to extract and amplify ancient DNA from insects trapped in amber, which directly inspired the premise of Jurassic Park.
And of course, years later, PCR was the exact technology used to create the billions of COVID-19 tests that helped manage the global pandemic.
So in 1993, Kary Mullis's contribution was officially recognized at the highest level. He was awarded the Japan Prize and the Nobel Prize in Chemistry.
The Nobel Prize.
Yes. And with that platform, he successfully cemented the narrative that PCR was entirely his solo invention, often refusing to credit the massive contributions of White, Arnheim, Ehrlich, and the rest of the CETUS team who actually made it work.
The story of Kary Mullis takes a very dark turn after the Nobel Prize.
Right.
But to tell the full story of the man behind this invention, we have to look at what happened next.
Mullis essentially stopped practicing traditional science. He used his newfound fame and Nobel laureate status to promote a series of highly eccentric and, in some cases, deeply harmful beliefs.
On the eccentric side, he wrote a book where he claimed to have encounters with glowing raccoons and believed he had been abducted by aliens.
He also started a company that used PCR to amplify the DNA of dead celebrities, like Marilyn Monroe, to sell as little trinkets.
But the darker turn came when he began using his scientific authority to promote outright denialism. He became a vocal denier of climate change. He denied the existence of the hole in the ozone layer. And most tragically, he became a prominent denier of the link between HIV and AIDS.
He publicly claimed that HIV was not deadly and that AIDS essentially did not exist. This wasn't just a fringe opinion spoken in a vacuum. His status as a Nobel winner gave these claims immense weight.
Yeah.
In the early 2000s, South Africa was battling a catastrophic AIDS epidemic. The president of South Africa at the time, Thabo Mbeki, fell under the influence of AIDS denialists and explicitly invited Kary Mullis to weigh in on the issue.
Relying on this denialist rhetoric, the South African government refused to provide adequate proven antiretroviral treatments to the public. That policy delay, influenced by figures like Mullis, led to the preventable deaths of over 330,000 people.
Mullis later passed away in 2019 at the age of 74 from pneumonia complications.
Synthesizing the man is incredibly difficult.
It really is.
Yeah.
On one hand, you have a mind capable of highly imaginative, playful brilliance. A mind that could visualize the invisible and conceptualize a tool that has undeniably saved millions of lives and advanced human knowledge immeasurably.
Yeah.
But on the other hand, that same mind was housed in a deeply flawed, abrasive, ultimately harmful personality.
So what does this all mean for you and me? Why does this story from the 1980s matter today?
Well, the deep dive here isn't just about a complicated scientist or the biology of an enzyme. It is a profound lesson about the nature of automation.
Exactly. Think back to Mullis in that lab before the Epiphany. He was basically a human pipette, spending all his time manually mixing radioactive probes. He only had the time, the mental bandwidth, and the materials to dream up this world-changing concept because a machine literally took over his tedious daily tasks.
That is a powerful reframing of a fear many of us have today.
Yeah.
We are currently staring down the barrel of an incredible surge in AI and generative models. There's a very real, very valid fear that machines are going to take over our jobs.
Right.
But the story of PCR suggests that automation doesn't just eliminate work, it eliminates busy work.
If our minds are trained in critical thinking and problem solving, Losing the repetitive parts of our jobs to AI shouldn't just be scary. It should be seen as the exact catalyst we need to free our minds to focus on the bigger picture.
Automation might be the very thing that gives us the time to stumble upon the next breakthrough that changes the world.
Which leaves us with a fascinating thought to take away from this deep dive.
Yes. If the automation of a single, mundane laboratory task in 1983 was the true catalyst for unlocking the entire human genome, what massive world-altering breakthroughs are currently trapped behind the busy work of today's greatest minds, just waiting for an algorithm to set them free?
That is an amazing question to chew on. Thank you so much for joining us on this deep dive into the messy, brilliant, and automated history of the DNA photocopier. Until next time, keep questioning, keep exploring, and we will see you on the next one. Farewell.